It's a privilege and honor to introduce Dr. Sharon Kujawa, who is a doctor and principal investigator at the Eaton Peabody Laboratories at the Harvard Medical School in Massachusetts Eye and Ear Infirmary. And she's kind enough to give us a talk today on putting the neural back in sensorineural hearing loss. So she's been I think what can we say, you and Charlie have been the founders of kind of hidden hearing loss, which is a big topic now. We're discoverers of that. So we appreciate you joining us today. So thank you.
Okay, well, thank you very much. And really thanks for the invitation to do this. Thank you, Dr. Bigelow, for the invitation to present this work. So I was asked, I think, to talk about our work on cochlear synaptopathy. And so, my focus will be here in an area that's well familiar to all of you. Basically, in the organ of Corti, where we have two subsets of hair cells that shape incoming information, where signals are communicated to auditory nerve fibers. They're transduced into a form that those auditory nerve fibers can use. And in my lab, we really spend a lot of time thinking about that communication between inner hair cells and afferent nerve fibers, and about the fibers that might be most vulnerable to the insults that we're focused on, which is noise exposure and changes that occur with aging. We're interested in who recovers on their own from those kinds of challenges, and who can be coaxed to recover with treatment, and of course the functional consequences of the loss when it is permanent. So as you all know, hair cells, particularly outer hair cells, are highly vulnerable elements in the organ of Corti. You can see, I think a lot of our attention has been focused in this area because the changes are so obvious and the consequences are reflected so well in physiology that we have access to. So of course, on the right, you see an audiogram obtained in life from an individual who had a significant noise exposure history. And then after donation of their temporal bones, you can see some of the underlying pathology, kind of focused outer hair cell loss, not so much inner hair cell loss for this particular example. So inner hair cells also, when they are lost, of course, interrupt communication, but in our hands, they're certainly much less vulnerable than the outer hair cells. And you have to lose a very large number of them to really substantially impact threshold sensitivity. So on the left is a panel from some experiments Charlie and I did actually when I was a postdoctoral fellow, with chinchilla model of carboplatin ototoxicity. So in the chinchilla, the loss is primarily of inner hair cells rather than outer hair cells. You can see that for this example, the outer hair cells are pretty much all there. The inner hair cells, there are regions where substantial losses are evident, but the thresholds for the compound action potential is falling within a normal range for our series of animals. Similarly, Edlow-Barinas originally from Dick Salvi's lab, looked at a behavioral model with the same drug with carboplatin, and you can see that in their series, almost about 90% of the inner hair cells needed to be gone before you really started to have a substantial impact on threshold sensitivity. So although we are often focused on, hair cell injury and loss, there are some interesting differences that hopefully will help us, work with underlying mechanisms. And even when hair cells survive, this work that Charlie Liberman did a few decades ago shows really elegantly that here with all these hair cells, kind of present at least, single unit thresholds from a noise exposure are elevated. And although the hair cells are there, sorry, getting my cursor here, you can see the corresponding injury to stereocilia, in this case greater in the inner hair cell stereocilia than the outer hair cell stereocilia, but fairly large permanent threshold shifts in this particular example, although all the hair cells are kind of there and accounted for. So not all exposures cause permanent threshold elevations, as you also well know. And my focus during this talk is really going to be on the neural side of things, the synaptic losses that commonly accompany the kinds of acquired sensorineural hearing losses that we study in my lab, which is noise-induced and age-related primarily. And my work in these areas has been primarily in models, in animal models. We are moving a good portion of some of this work as we're able to humans. And so I hope to be able to share some of that with you later in this talk. So for me, the questions really began with an interest in a long outstanding question. That is, does a history of noise exposure change the way ears and hearing age going forward? And when I started this work, our focus, like almost everybody's at that time, was on PTS producing exposures, shown up here in this graph. This is an observation that has been made over and over again in many labs, where at short post-exposure times, threshold elevations are largest. They kind of recover with an exponential time course. And then in the case of a permanent threshold shift producing exposure, of course, they stabilize at a level that is elevated relative to the pre-exposure baseline. And how much they are elevated, how fast and how completely they recover is related to many factors of the exposure in the frequency and time and level domain. It's also related to vulnerabilities of the individual. And we don't really have time to talk about all of those elements today, but I'm sure you're all well familiar with that. In the case of temporary threshold elevations, of course, you see this kind of more rapid recovery of threshold sensitivity. And by design, these exposures are temporary. In other words, threshold sensitivity comes back to a baseline level after time, sometimes hours, days, and even up to a couple weeks before these thresholds will completely recover. In the case of the permanent threshold elevations, this stabilization here has really been taken as evidence for no delayed effects of noise on the ear and hearing. And that was kind of the dogma that we faced when we were starting to work in this area. And in the case of the temporary threshold shifts, of course, this recovery to baseline was really taken as evidence that the exposure wasn't really all that bad. It was pretty benign, and that you now had a recovered ear from the exposure that was experienced. So we decided to test this directly, and we did that because of some preliminary information we got in a retrospective analysis of human data from the Framingham heart study cohort. Those individuals had been studied over decades, and there were some indications that for men with a history of noise exposure that was reflected as audiometric notches, that the changes that they experienced as time went on were different. They were different from those that didn't have that audiometric kind of signature of a noise notch. And, you know, there's a lot of hand wringing associated with an observation like that, because of course it wasn't designed, the study was not designed to really carefully assess the noise exposure of these individuals. And there was no control, of course, of all of the other ongoing exposures they might have had after they retired from their working lives. And so we decided to test this directly in an animal model where we could control the genetic background, we could control the age, the exposure, we could deliver just one intended exposure, and then know, you know, even though you can't take all exposures away in an animal care facility, we can definitely quantify them and then compare them to animals that are otherwise held identically, but without the single exposure. We tested outer hair cell-based distortion product emissions, neural-based ABR responses. We see the threshold elevations 24 hours after an exposure. This is our exposure band that we used. So in mouse, that falls right about in the region where their threshold sensitivity in the normal case is best. You see these large acute elevations and then a return to baseline in the weeks following the exposure. Distortion product amplitudes also recovered. Here you see 24 hours, but in the weeks following, they really don't look different from animals that never received the exposure. But of course, the observation that generated our interest and subsequently the interest of others is this persistent decline in the neural response. And this is a huge decline because this is a log-based scale here. So following up on this observation, we looked at the tissues from these ears. This just basically shows you the region of our focus here. So in the cartoon on the left, the synaptic communication between the inner hair cell and the auditory nerve fiber. You know that each of these fibers makes a single synaptic contact with a single inner hair cell. This is kind of what it looks like in the neural pill underneath the inner hair cells themselves, and a blow up here of one that I've circled. So with a loss of this communication, of course, that means that this auditory nerve fiber is completely silent because that's its only input. So having the means to be able to quantify, because the immunostains make these tissues proteins of interest that are visible to us, we can then use methods to pair the presynaptic ribbon with the postsynaptic element. Here we're looking at a glutamate receptor patch that is opposed across the membrane to that presynaptic ribbon. And then it's a simple means then to just count the dots, basically. And so when we do that, we're able to quantify the changes that we see compared to a normal ear. So here on this graph, you see number of synaptic ribbons per inner hair cell in a control group of animals. And then after the noise exposure that I just described to you, you see this dramatic reduction in the number of synapses. And for this particular exposure, in this particular strain of mice, these were permanent. You can see that 3 days, 8 weeks, we subsequently held these animals to the end of their lifespan. And basically, these did not recover. There's some jumping around at early post-exposure times because the proteins become internalized in the membrane and the stains are not so sensitive to detecting them. But when things stabilize a few days after the exposure, you're left with these pretty robust reductions in the number of synapses that you have. So we took the opportunity, a couple years ago to take all of the exposures that we have done in our mouse model and just compare them using a metric that, frankly, the Occupational Safe and Health administration uses to kind of titrate human exposures on the job. And so it's a time-weighted average where you basically look at the amount of noise dose that an individual gets over a specific period of time. For OSHA, that's typically an 8-hour workday. Here, we just titrated everything along this time-weighted average axis, and then first looked at the permanent threshold shift in the ABR wave 1 at 2 frequencies, a frequency that was not so involved with our noise exposure, and one that was maximally involved. And what you see is, for a really broad range of exposures that we have used, permanent threshold shifts were not common for these exposures. It wasn't until past about 90 dB that we started to see threshold elevations. And of course, if you go higher or longer, this is going to shoot up and you will end up with whatever level of threshold elevation you get with these really extensive noise exposures. But in the same ears, we looked at the synapse counts and pretty dramatically show here that even, before you start seeing changes in threshold, permanent changes in threshold sensitivity, the synapse loss has pretty much maxed out. We rarely see any conditions of the noise exposure where we get more than about 50, 60% synapse loss. And so here things are starting to stabilize at that maximum. The graph is shifted to the left, and you can see dramatic synaptic loss in these same ears where you basically have no audiometric evidence at all that there is any ongoing injury. So we have started to look as well at mixed neural and sensory losses. This obviously will have important clinical, applications, because people who walk into the clinic, of course, don't necessarily or even commonly have strictly neural loss. And so there will be different levels of hair cell involvement. And that is an issue that is, of course, the most challenging, I think, to address in the clinic. Let's see, why did that happen? Okay, so, oops, again, what we're doing is looking at a range of noise exposures. And in this case, I used equal energy exposures. So basically trading time and intensity to create noise exposures that intend to give equal energy. For this range that we're showing here, you can see fairly mild permanent threshold shifts restricted to basal cochlear frequencies for this series. In these ears, there was essentially no inner hair cell loss to speak of. And in the extreme base, some of the exposures produced some outer hair cell loss. There was one highest level but shortest duration exposure that produced the most outer hair cell loss. But again, in the same ears, you see this dramatic reduction in synapse counts in the region above the noise exposure band. Looking at this instead of equal energy exposures, but equal levels. So to keep the area that we were injuring in the cochlea kind of constant, we just had a 100 dB exposure, and then we just changed the duration of the exposure. Again, you can see some outer hair cell loss in the basal region of the cochlea, but really a nice dose responsive loss of synapses in the same region that we saw the threshold elevations after the noise exposure. So observations like this, are basically taken at fairly short post-exposure times, a couple weeks after noise. And getting back to really my original interest in this area was, well, what happens if you noise expose and then you just hold those animals for a long post-exposure time and not give any additional intended exposures? And so in another series of experiments, that's exactly what we did using the same animal model. This is the model. It's a CBA-CAJ mouse. And so it looks from graphs like this that the really functionally important structural changes appear to be focused, on outer hair cell injury. You see here that not much is going on as age approaches, middle age. It really isn't until about middle age where outer hair cell counts start to fall, and they fall pretty dramatically beyond that point. Inner hair cells stayed fairly robust throughout the entire range of ages that we monitored. So 140 weeks is pretty old for a mouse. And then we looked at distortion product changes over the same range of time. And you can see that those mirror relatively nicely the condition of the outer hair cells in the same ears. So distortion products are normal, normal, normal until you start having outer hair cell injury and loss, and then they start to elevate as well. All right, my cursor is not wanting to move. Okay, so in these animals, then we also quantified synaptic losses. And you can see that in contrast to the kind of late changes that you see for the hair cell injury, synapse counts just march progressively downward over the course of the entire lifespan. We see it throughout the cochlea. So for aging, that's in contrast, of course, to what we see for noise exposure because we're targeting a region of the cochlea. But in our aging model, you see, this gradual decline in the number of synapses per inner hair cell throughout the cochlea. If you look again at that region where thresholds and hair cells remained quite normal, you see that you're already at about 20–25% loss of synapses in that middle age. So what happens then to address my original question? What happens if animals are noise exposed first and then aged? And so we do that, and then we hold them with never exposed animals and compare outcomes. And what you see in the panel on the right is just the gradual decline in synapses at 32 kilohertz and at 11 kilohertz in the unexposed animals. After noise, there's an immediate, for this exposure, roughly 50% loss of synapses, which persisted and stayed at about that level, 50 to 60% throughout much of their lifespan. In a region where we really had no evidence of initial synapse loss with the noise exposure, so at 11.3 kilohertz, there wasn't any synapse loss. What you see then as age marches on is this separation of age alone versus noise, then age animals, to where you're starting to see exaggerated synapse loss in regions that did not appear at any rate to be initially targeted by the noise exposure. If we look at ganglion cell counts, so of course in the human histopathology world, a lot of this has concentrated on quantifying ganglion cell counts. Here in the mouse model, what you see is and I apologize, these sections are thick enough that you can't really even resolve the individual cells in the region of the spiral ganglion. But in a young exposed animal, of course, you have a very robust population of the spiral ganglion cells. For just aging alone, you see some dropout at I think that quantifies to be about 35% in a 2-year-old mouse in our strain that we used. But if you noise expose the animal at 16 weeks and then hold it to 2 years, what you see is this dramatic decline. This is kind of a late development, so this doesn't happen immediately. You know, it takes months for these cells to really start to disappear. And then at advanced ages, of course, you can see the dramatic effects of that prior noise exposure. But analyses like this really don't tell us anything, at least the way we did them at this time, about who died, right? So we don't really know much about the subtype targets of noise exposure or of aging in our animal model. And that's something that we're spending more and more time on, because of course that should have important functional consequences, because fibers have different properties of response. So what we have started to do is basically look at vulnerability of different subsets of auditory nerve fibers based on their spontaneous rate categories. And so just basically reviewing that briefly for you, high spontaneous rate fibers are more sensitive. They're the ones that really determine our threshold sensitivity. So they have good thresholds, and they have very strong onset synchronization. This onset peak is everybody, you know, firing in registration, and then this adaptation to a steady state level of firing. So in these kind of post-stimulus time histograms, you will see this type of onset response followed by this temporal change in firing rate. Medium spontaneous rate fibers, of course, are intermediate in these ranges. I'll show you here now on the left, low spontaneous rate fibers. And what you see is elevated thresholds compared to higher or medium spontaneous rate fibers, and then rate intensity functions that continue to grow with increases in stimulus level, but which have very reduced onset synchronization. There's a fair amount of jitter in the onset of these responses, and they are a little bit later. And so it's like they have less onset response, but the plateau region is pretty similar to what we see for the high spontaneous rate fibers. You can also see a distribution around the inner hair cell in terms of the general locations where these fibers synapse with inner hair cells. And so Charlie Liberman has shown very clearly in several animal models, kind of, you know, that high spontaneous rate fibers versus low spontaneous rate fibers tend to cluster on different sides of the inner hair cell. And so that is also an assay that we try to use to help us differentiate, you know, who died and who's coming back and so on. So the reason that this really becomes important is that when you add a noise background, a number of labs have shown that, you know, here in quiet, these high spontaneous rate fibers are very sensitive, but then, you know, they reach this firing plateau, and increasing levels of stimulation really don't drive them to much higher firing rates. If you put them in a background of noise, however, they just get saturated by the noise. And so they don't continue to code information as you change level of the target stimulus that you're looking at. In contrast, low spontaneous rate fibers do a fairly good job of that. They have elevated thresholds, but they are able, even in backgrounds of noise, to continue to increase their firing rate in response to increases in the level of the stimulation, which suggests then, and has suggested to many people, that the high spontaneous rate fibers are important at threshold. So they're important in quiet, helping us hear out things at the threshold of sensitivity. whereas low spontaneous rate fibers may be more important in supra-threshold conditions, for example, speech in a background of noise. And so that is something that has led people to suggest, well, maybe, if you lose your low spontaneous rate fibers, that would be something that could underlie speech and noise difficulties, and it would not have to be accompanied by a threshold elevation, because these guys have bad thresholds to begin with. And so if you lose them, you won't really miss this in the threshold sensitivity. Where you will miss it is in supra-threshold signal processing. So, having that kind of motivation to really look at differences among the auditory nerve fiber subtypes, we have moved some of our work to the Gerbil. And so the Gerbil is a lower frequency hearer, which is also relevant to human, speech processing. But in addition, there's quite a bit known about the distribution of auditory nerve fibers as a function of spontaneous rate in the Gerbil. And in the left panel here, you can see, as a function of cochlear frequency and coded by color, you can see a lot of the high spontaneous rate fibers in red are clustered in kind of more apical to mid-cochlear regions. You can see that quantified over here. So high spontaneous rate fibers are in red, and then medium, and then low in green. And so there are fewer, of course, low spontaneous rate fibers, but they tend to mostly occur at more apical cochlear regions. And so this gives us kind of a model where we can target, knowing their distribution in the normal cochlea, we can kind of look by designing the experiments to look at different regions of the cochlear epithelium. And that is work that we have been partnering with a group in Montpellier. So Jerome Bourrienne has done this work here with single unit recordings in the Gerbil model. And then we are working very closely with Jean-Luc Puel as we study not only fiber losses associated with noise exposure and with aging, but also with glutamate excitotoxicity relative to cochlear application of the agonists themselves. So just as an example here, this is not a glutamate agonist, of course, but using the neurotoxin wabane, Jerome did a series of studies where he titrated wabane dose to look at, who drops out first, basically. And the results of this kind of dose response experiment is that low spontaneous rate fibers were indeed the most vulnerable to the wabane insult, followed by medium spontaneous rate fibers, and then high spont fibers in these animals. Okay, why is this not working? Okay, and so synapse counts in the same ears, again, show the clustering of synapse losses in the more basal regions of the cochlea, suggesting that this is targeting more low spontaneous rate fiber loss and synapse loss than high. In these animals, compound action potentials were recorded at the level of the round window. And surprisingly, what you see is not much, right? So the control animals and the wabane treated animals had compound action potentials that were virtually identical across a range of frequencies in the cochlea. And thinking about what we've shared, talking about single auditory nerve fibers PSTH kind of response properties, you can see why this might be the case. So these compound action potentials are really mostly reflecting an onset response, and that onset response is dominated by the high spontaneous rate fibers. So what this implies is that the persistent changes that we're seeing in the Wabane treated ears, the ones where the low spontaneous rate fibers were most sensitive, is not going to be well reflected in the suprathreshold amplitudes of these compound action potentials. So what we've started to do in working with our colleagues in France is to develop a gross potential that can be recorded at the level of the round window in the gerbil, and which follows this temporal course that is evident that you saw in the PSTHs. And so they have named this response the post-stimulus time, peristimulus time response. You can see that its onset, peak, and plateau regions pretty much mirror what you would see as the sum of all of the individual fiber responses in these ears. And of course, the reason to move towards this is not only are single unit experiments exhaustive, but they're not going to be applicable to human patients. And so we're trying to move some of these means of gaining this information. You know, right now, everything that we're collecting in the clinic, the ABR wave 1, and, you know, by electrocochleography, basically the compound action potential, are all onset responses. So they are all looking primarily at high spontaneous rate fiber activity, and they're not very well able to reveal losses of low spontaneous rate fibers. Whereas this response that we are working on in our animal models is sensitive to different types of fiber loss. So in this example here on the left are PSTRs that we recorded in unexposed gerbils here at 4 kilohertz and at 16 kilohertz. So looking at the right, you can see the distributions of low, medium, and high spot fibers in those two cochlear regions. And then we also studied them in animals that had received a noise exposure, and we looked at these responses either at 2 weeks or at 4 weeks post-exposure. And you can see that in the 16 kilohertz region, where you have a combination of low, medium, and high spot fibers, you're able to see the reduction. I appreciate that it's really hard to see the reduction at the level of the peak. I should have put some arrows there. But in the plateau, you can clearly see the reductions at 2 weeks and a little bit of recovery by 4 weeks of the plateau region of these responses. If we express this, you know, kind of as a growth function of stimulus level in the bottom right panel, what you can clearly see is the persistent reduction in the level of the plateau in these noise exposed animals in the 16 kilohertz region, whereas the peak responses were less affected in these animals. At short post-exposure times, they would have been declined, but then you would have had the combination as well of outer hair cell injury. So the persistent changes in these animals were primarily in the plateau, reflecting what we suggest is a persistent reduction in low spontaneous rate fiber counts, whereas high spontaneous rate fibers either were less vulnerable to the insult or recovered rather quickly after the insult. in the noise exposed animals, we also measured spontaneous activity recorded at the round window. So people have used round window noise recordings for a long time. This, in our animal model, you can see that 24 hours after our noise exposure, which is a TTS producing exposure, you know, this exposure produced no PTS in our gerbil model. But you see the dramatic reduction in the magnitude of that 900 hertz component of the spontaneous round window noise, and then recovery after. Again, this should reflect primarily high spontaneous rate fiber activity, just due to the sheer numbers of the fibers that the round window recording has access to. Remember, this is not a stimulated condition. So we've also started to look at these properties of nerve activity in our aging gerbils as well. You can see both in the spontaneous response that we record at the round window and in the stimulated response, the PSTR, in the same animals, you see a pretty obvious age-related decline in the magnitudes of these responses. So in the case of the aging model, not only in the onset response or in the kind of high spontaneous rate fiber dominated responses, but also in the region of the plateau as well, which should be reflecting the contributions of all of the fiber subtypes. So what about humans? You know, human work is something that we are starting to be more and more involved in. Certainly my colleagues have been doing parallel investigations in human patients and in subjects who really have no known auditory pathology, but may be high risk for noise exposure, for example, musicians. And so what I'd like to do is just kind of share with you some of the things that we have been doing so far in comparing what we see in the animal models and in human patients. And in this graph, several years ago, Charlie and I took the opportunity to kind of compare what we see in the animal models versus the temporal bones that he has been studying at the Mass Eye and Ear. And you can see in the black and in the red symbols, the synapse survival in our animal models as compared to the terminal. So he's looking at terminals primarily because they are more robust to the processing. But you can see this dramatic difference as the age of these individuals is expressed as percent lifespan. And so something that you know is intriguing to think about is this noise. You know, we control very carefully the exposures that our animals get over the course of their entire lifespan in the animal care facility. We monitor our sound levels in the rooms for our animals, and we have a pretty good idea about what they are exposed to as unintended exposures. And we also have animals that, you know, are being exposed versus non-exposed in that way so that we can compare. Whereas humans, of course, you know, a lifetime of noise exposure, you know, may be reflected in these kinds of counts. We never can really know from these human temporal bones exactly what somebody was exposed to over the course of their lifetime, noise or not. But certainly that's one important difference between the three groups that are shown there. So Charlie started to look at that a little bit more explicitly in the human temporal bones. He now has a number of cases where a noise exposure history is documented, and looking at cases in that way, it becomes clear that in In cases where there is a documented history of noise exposure, that the exposed individuals have greater outer hair cell loss, have greater auditory nerve fiber loss, and poorer audiograms as they age. I think the compression that you see at the oldest age span is probably because the targets of the noise and the aging overlap. And as George Gates used to say, you can't kill a hair cell twice. So if it's lost from noise, it's not going to be lost from aging as time goes on. So, there's been a lot of hand wringing, I think, about whether this is a big deal in humans. And certainly there are huge differences in susceptibility between humans and animal models. We know that the animal models differ from each other on measures of their susceptibility to any number of things. But there are large numbers of humans who are exposed to very high levels of noise. And even the fairly benign things that you're talking about, so I was involved in a NIOSH funded study a number of years ago where we looked at construction workers and tried to quantify the kinds of exposures that they would routinely get on the job. And you can see that if you follow OSHA guidelines for exposure, that not only are many of these workers getting these exposures without ear protection, but they are getting them for far longer than the doses that are allowed by OSHA for exposure. And the construction industry is an unregulated industry with regard to noise exposure. It's kind of an important side point, I think. But then you look at exposures like this, and you all who maybe have military populations close to your clinics, and you see some of these people, they are being exposed to unbelievably high levels of sound. Even with double ear protection, they are really in an untenable situation for receiving noise exposures that are surely synaptopathic. So how are we going to know? It seems like our metrics that we have in the clinic are non-specific. They're not very sensitive. They're contaminated often by, not contaminated, but complicated in interpretation by hair cell based injuries. And so, we have things like auditory brainstem responses where we can capture overlapping information with what we get from the animal models. We can refine that a little bit and do electrocochleography and get bigger responses closer to the generators. Again, these are all going to be most sensitive to high spontaneous rate fiber status as the onset responses. And so now we are working with a number of investigators at different sites to start moving this work to humans. And so this post-stimulus time response is now being investigated as a way for us to learn more about human auditory nerve function and dysfunction. So just as a summary, you see here the PSTRs recorded in our Gerbil model at 2 different frequencies, looking at peak to plateau ratios. So you can see you measure the peak response value, the plateau, and generate these curves of response. And similarly in human patients, where we are taking advantage of opportunities in the operating room where we have access to auditory nerve recordings. This series of 8 patients was done in France with our colleagues. So these individuals had normal thresholds. Compound action potentials were recorded in response to clicks. and PSTRs were recorded at a couple different frequencies. And so it's just kind of a first demonstration of feasibility of some of these response recordings. We're now getting underway to look at these in a larger series through several clinics to try and take this information and learn more about human auditory nerve function, where we have no idea what the distribution of fibers is, low, medium, and high spontaneous rate in the human auditory nerve. So our approach here is basically multi-pronged, because we know we can't address all of the questions with one technique. And so we use our animal models where we can know, we can measure the function, we can know the underlying histopathology, we can create targeted lesions, in this case, a Wabane dose that basically removed most of the neural response here, and what you're left with in red is primarily a summating potential, which also has some neural components in it, but it certainly has hair cell components in it as well. So we have opportunities like that with our experimental manipulations. In human temporal bones, we can do parallel immunostaining to get these beautiful images that Charlie is now getting from human temporal bones. It's really the only direct way that we have at this point to know what these synapse counts are in the human ear. And again, pairing that with the traditional kinds of hair cell counts and of course the pathophysiology in the same individuals. Now moving forward, using these techniques that we think will be more informative to subtypes of neural loss, so that we can see who's most vulnerable to these insults, who recovers on their own, who can be coaxed to recover with treatments, and then what the functional consequences are of the loss. And pairing that as well. So we take the human data, we take the animal data, feed it into in silico models that Jerome Bourrienne is developing, so that we can additionally translate some of this information to inform what we think is going on in the human participants. So these are the individuals that have contributed to all this work. It obviously takes a great range of expertise and my funding agencies. And with that, thanks for your attention.
Hello, my name is Dr. Kevin Peng, neurologist here at the House Institute. Thank you for watching this video. The House Institute provides free educational videos for hearing health professionals worldwide. To help support videos like these and other educational efforts, please consider donating by clicking the link in the description box below. Your generous support allows us to keep videos like these at no cost for you and others. Thank you.